Welcome to the Single-Cell Codex. This is Chapter 1, and it answers two questions. What is single-cell RNA sequencing, and why does analyzing it require computation? Here is the line the whole chapter turns on. The table arrives looking like a fact. It is a residue. In plain language, the neat spreadsheet of numbers you receive is the downstream result of many experimental and computational decisions, and most of them happened before you opened the file. Learn to read what those decisions left behind, and everything after it gets easier. Let us start with a plain definition, before any of the nuance. Single-cell RNA sequencing measures captured RNA molecules from individual cells, or from individual nuclei, allowing researchers to identify cell types, cell states, and condition-associated changes within complex samples. Note the word captured. You are not measuring every molecule in the cell. You are measuring the ones the experiment managed to capture and sequence. Hold on to that word, because most of what follows in this chapter is a consequence of it. So why do it at all? Biology has always been about validation. Two cells that look identical under a microscope can behave differently, respond differently to a drug, or head toward different fates. For decades our methods forced us to ignore that, because in bulk RNA sequencing you extract RNA from thousands or millions of cells at once and the data are an average signal. If a gene is highly expressed in a small subset of cells, that signal gets diluted, and bulk cannot tell you whether two genes are switched on in the same cell or in different cells. Single-cell sequencing makes that hidden heterogeneity observable. Each cell is treated as its own experiment. But be careful how strongly you state the win. Single-cell data reveals heterogeneity that bulk averages conceal, and it introduces new sources of uncertainty of its own: shallow sampling, doublets, ambient RNA, and imperfect cell classification. It trades one blind spot for a different set of them. And it would be easy to read that as an obituary for bulk RNA sequencing. It is not. Bulk remains the correct choice for a large class of questions, and reaching for single-cell out of novelty rather than need is its own kind of error, more expensive, noisier, and often worse at answering the actual question. The reason is statistical. Bulk measures each gene deeply in every sample. A single-cell experiment spends the same sequencing budget across thousands of cells, so each cell, and each gene within it, is measured shallowly. If your question is genuinely about the average, measuring the average directly is usually more precise than reconstructing it from thousands of sparse, noisy per-cell estimates. Now, what actually happens experimentally? Strip away the chemistry and the experiment does one thing. It partitions a tissue into cells or nuclei, tags each one's RNA with a barcode marking where it came from, sequences everything together, then sorts the reads back out by barcode. It is worth tracing the path once, because every arrow discards something. Tissue, to dissociation, to barcoding, to library preparation, to sequencing, to alignment and counting, and finally the count matrix. You will probably not run the early steps yourself, but you inherit every choice they made, which reference genome, which counting rule, which filtering threshold, before you ever open the file. Two families of protocol dominate, and they trade the same currency in opposite directions. Plate-based methods, like SMART-seq2, sort individual cells into wells and sequence each one deeply, across the full length of a transcript. That gives greater sensitivity to splice junctions and transcript isoforms than a standard 3' droplet assay, on hundreds of cells. Droplet-based methods encapsulate cells in barcoded nanoliter droplets. You get tens or hundreds of thousands of cells, each sampled shallowly, usually only at one end of the transcript. Neither is better. To count how many cell types are in the cortex, you want breadth and you accept shallow sampling. To study transcript structure in a rare population, you want depth and you accept fewer cells. One piece of the measurement deserves attention because it is invisible in the final matrix. Between capture and sequencing, the cDNA is amplified by PCR and amplification is not uniform. A molecule that happens to amplify well produces many reads, an identical molecule that amplifies poorly produces few. So if you simply count reads, you are partly measuring amplification efficiency rather than abundance. A unique molecular identifier is a short random tag attached to each molecule before amplification. All the reads descended from one original molecule carry the same tag, so collapsing reads by tag recovers a count of molecules rather than a count of amplification products. State the benefit carefully. is reduced PCR amplification bias, which makes counts more proportional to captured transcript abundance. They do not hand you every molecule the cell contained. And note that SMART-seq2 has no UMIs, so its counts mean something different. SMART-seq3 added them. When you read a method section, find out which. So here is the object you actually receive. Rows are genes. Columns are cells, or, in brain tissue, usually nuclei. In UMI-based assays, each value is the number of captured transcript molecules assigned to a gene in a cell. Look at a small slice from human cortex and two things appear at once. First, identity. One nucleus shows a pan-neuronal marker plus an excitatory marker, another, the same pan-neuronal gene plus an inhibitory one. A third shows an astrocyte marker, a fourth, an oligodendrocyte marker. Second, and this is the part beginners miss, the zeros are not simply noise obscuring the signal. The pattern of zeros carries much of the information about which cell is which. Before we go further, we need to separate 5 things that beginners routinely blur together because a great many statistical mistakes in this field come from confusing them. A molecule is one captured transcript. A gene is expression summarized across transcripts. A cell is one column in the count matrix. A sample is one biological specimen containing many cells. And a donor, or animal, is the biological replicate you actually use for inference. That last distinction is the one that matters most. Thousands of cells from 3 donors give you information about 3 donors, not about thousands of independent observations. Cells within a donor are not independent biological replicates, and treating them as if they were is the most common way single-cell results get inflated. Now to the property that surprises everyone the first time. Single-cell count matrices are extremely sparse. In many droplet-based datasets, more than 90% of matrix entries may be 0. In the Allen Brain Cell Atlas Cortex subset used in this book, roughly 92%. But that figure is not a universal constant. It depends on the protocol, the tissue, the sequencing depth, and how many genes you kept. Let us see why the zeros are expected rather than broken, and to do that we need to be careful with vocabulary, because 4 different quantities are easy to confuse. Suppose a nucleus expresses about 10,000 distinct genes. Those genes are represented by a much larger pool of individual transcript molecules inside the nucleus. A droplet protocol captures only a fraction of that pool, on the order of 10%, so after sequencing you might observe roughly 2,500 captured molecules, that is, distinct UMI tags. Those captured molecules are spread across genes in proportion to abundance, so the number of genes actually detected ends up far smaller than the number expressed. Most genes sit at only a handful of molecules. Draw a small sample from a pool dominated by a few abundant transcripts, and many of the rarer ones simply will not appear. Not because they are absent, but because you did not draw them. So what does a 0 mean? Most zeros reflect limited sampling. The gene was expressed, but you did not capture any of its molecules at this depth. Some zeros may also reflect true biological absence, where the gene really is off in that cell. The honest position is that you usually cannot tell which is which from the matrix alone, and that is exactly why the distinction matters. This is also where beginners reach for imputation, which fills in the unobserved values. Be careful there. Imputation estimates unobserved expression, and used without careful validation it can distort biological structure, smoothing away real differences or creating apparent co-expression between genes that were never co-expressed. It is a modeling decision with consequences, not a cleaning step. One more experimental choice that shapes every threshold downstream: cells or nuclei. In single-cell sequencing, an intact cell is captured and lysed, and RNA from both the cytoplasm and the nucleus is sequenced. In single-nucleus sequencing, the cell is broken open, the nucleus is isolated, and only nuclear RNA is captured. The cytoplasm, and most of the mature messenger RNA it holds, is left behind. The two produce matrices of identical shape and different meaning. The practical rule is mostly about the tissue. If it dissociates cleanly into viable cells, such as blood, dissociated tumors, or cultured cells, single cell recovers more RNA per cell. If the tissue is frozen, patty, or fragile as adult brain typically is, nuclei are often the more honest option. Which brings us to the idea that reorganizes everything else. Sequence the same library twice as deeply, and you detect more genes per nucleus. Sequence it half as deeply, and you detect fewer. Nothing about the underlying biology changed. So stated plainly, the count matrix is a noisy, incomplete measurement of a biological sample. Every entry is better understood as a random variable than as a fixed property of that cell. What you hold is one realization of a process you never observe directly, and every quantity you compute from it, genes per cell, a fold change, a cluster boundary, is an estimate that carries uncertainty and moves when the depth or the threshold moves. If the value in a cell is a random variable, what is shaping it? At least these. The cell's true biological expression, which is what you actually want. Real biological variation between cells and between donors. Then the whole sampling and technical process, capture efficiency and amplification noise, the sequencing depth you happen to allocate, ambient RNA leaking from lysed cells, doublets, where two cells shared a barcode, dissociation stress that changes expression before you even measure it, and differences in total RNA content between cells or nuclei. And finally, the computational choices, which reference genome, which counting rule, which filter. Only the first group is biology. Everything else is process. And you cannot separate them by staring at the matrix. You separate them with experimental design and with statistics. So what do researchers actually do with this matrix? 6 things broadly. Identify cell types, which is the census of what populations are present. Discover cell states, which are transient conditions such as activation or stress rather than stable identities. Compare cellular composition between conditions. Did a population expand or shrink in disease? Test gene expression changes within a population between conditions. Reconstruct trajectories, cautiously, to order cells along a process such as differentiation, and cautiously is the operative word, because an algorithm will happily draw a trajectory through data that has none. And map cells onto reference atlases, transferring labels from published data. Almost everything in the rest of this book serves one of these 6 tasks. Underneath those tasks, almost every single cell study is really asking one of 3 questions, and naming them gives each new method a place to land. Discover what cell types and states are here. That is clustering, annotation, atlas building, and its characteristic risk is calling something a new cell type when it is only a cell state, a technical artifact, or a clustering parameter. Compare what differs between conditions. Disease versus control, young versus aged, which populations shifted and which genes moved. Its characteristic risk is the pseudo-replication trap, mistaking cells for replicates, exactly the confusion we drew out earlier. And map, where does the cell belong? Placing new data against an existing reference, with a risk of forcing a confident label onto a cell that has none. Knowing which of the 3 you are asking is what tells you which method is appropriate, and which risk to watch. Which brings us to the second question in the title. Why does this require computation? 3 reasons. Scale. The datasets are far too large to handle by hand, and the operations you need. Normalization, dimensionality reduction, clustering, differential expression, require explicit, traceable computational decisions, whether you execute them through code or through a graphical interface. Structure. When you filter out a low-quality cell, the matrix, the annotations, and the embeddings all have to update together, which needs structured objects that travel as one. And reproducibility. Every choice, the mitochondrial threshold, the number of principal components, the clustering resolution, shapes the result. Scripted workflows make those decisions easier to reproduce, review, and audit. That is the real argument for code, not that a graphical tool is forbidden, but that your decisions should be recorded somewhere another person can check them. And if you want the scale claim demonstrated rather than asserted, here it is. The cortex subset used in this book has 40,000 nuclei and 32,285 genes. A modern Excel worksheet holds 16,384 columns. Put genes as columns, and 32,000 exceeds that limit by a factor of 2. Put genes as rows instead, and 40,000 nuclei exceed it by 2.5. So the object does not fit in a conventional worksheet before a single analysis is run on a deliberately modest subset. And the deeper problem is not only the dimensions. A flat table stores every 0 explicitly when more than 90% of them are 0, and it has nowhere to record the metadata, the embeddings, or the uncertainty that travel with the data. A last warning before the methods begin. Modern single-cell analysis is dominated by powerful pipelines. Cell Ranger turns reads into a matrix. scRNA and scRNA take that matrix to publication figures in remarkably few lines. That accessibility is a genuine achievement. It is also a risk. When analysis is easy, scrutiny becomes optional. Parameters accepted by default, outputs trusted without inspection, visualizations read as truth rather than as projection. Every one of these tools encodes dozens of default choices, filtering thresholds, normalization schemes, neighborhood sizes, and a default is a decision someone else made about a dataset that was not yours. So treat tools as instruments, not authorities. Instruments you inspect, calibrate, and question. So, back to where we started. Single-cell RNA sequencing measures captured RNA molecules from individual cells or nuclei and lets you identify cell types, cell states, and condition-associated changes. What arrives on your disk is a count matrix that is sparse, noisy, incomplete, and shaped as much by the process that produced it as by the biology inside it. The table arrives looking like a fact. It is a residue, the downstream result of many experimental and computational decisions. The rest of this book is about reading it honestly. Keep the uncertainty in view. Remember that the donor, not the cell, is usually the unit of inference. Know which of the three questions you are asking. And treat your tools as instruments rather than authorities. The full written chapter, with the worked examples and exercises, is on multiomeacademy.com. In the next chapter, we start turning this matrix into biology. See you there.